Have you ever wondered why populations that live close to one another often share similar characteristics, even if they started out quite distinct? Or how a new beneficial trait might spread across a wide geographical area, seemingly defying isolation? The answer often lies in a fundamental evolutionary process called gene flow. Gene flow refers to the transfer of genetic material from one population to another, typically through the movement and subsequent reproduction of individuals. Its primary effect is to introduce new genetic variations into a population or alter the frequencies of existing genetic variants, also known as alleles, thereby directly influencing the genetic makeup and evolutionary trajectory of those populations. At its core, gene flow works by transporting genes. When individuals migrate from one population and successfully breed within a new population, they bring their unique set of alleles, which are different forms of a gene, into that new gene pool. This genetic exchange has several profound effects. Firstly, it tends to reduce genetic differences between populations. Imagine two populations that were once isolated and began to diverge genetically. If gene flow begins, the continuous exchange of genes will make their genetic compositions more similar over time, leading to a process known as genetic homogenization. Secondly, gene flow can introduce entirely new alleles into a recipient population, significantly increasing its genetic diversity. This influx of new genetic material can be incredibly beneficial, potentially providing the raw material for adaptation to changing environments. Conversely, it can also spread maladaptive traits if migrants carry genes that are detrimental in the new environment. Finally, gene flow can counteract other evolutionary forces like genetic drift and natural selection, either preventing populations from diverging too rapidly or slowing down local adaptation by continually reintroducing less adapted alleles. Scientists observe and understand these effects by meticulously studying population genetics. One common approach is to compare allele frequencies, or the relative proportions of different alleles, in various populations over time. Significant changes or similarities in these frequencies, especially between geographically distinct groups, often point to the occurrence of gene flow. Researchers also track actual migration patterns of individuals and use sophisticated genetic markers to trace the origin and spread of specific genetic traits across landscapes, providing direct evidence of genetic exchange. Gene flow is not just a theoretical concept. We see its effects in the natural world through phenomena like hybrid zones, which are areas where two genetically distinct populations meet and interbreed, producing offspring of mixed ancestry. Another fascinating manifestation is clinal variation, where allele frequencies gradually change across a geographical gradient, often due to continuous gene flow between adjacent populations. For example, human skin pigmentation shows clinal variation, gradually lightening with increasing distance from the equator, partly influenced by ancient migrations and gene flow. Understanding these patterns helps us piece together the complex history of populations and their interactions. Ultimately, gene flow acts as a powerful connector, intricately linking populations across landscapes and generations. By facilitating the movement of genetic information, it plays a critical role in shaping genetic diversity, influencing the adaptive potential of populations, and dictating the extent to which distinct groups remain genetically interconnected or diverge. It is a dynamic force that continuously reshapes the evolutionary landscape.